Hi friends, welcome to today's section. So let us discuss about the cell organelles such as lysosomes, endosomes and microbodies. Myself, Jumana Karadan, 2nd MSc Biology from St. Mary's College, Thrissur. I am submitting in the guide of Dr. Anu P.A., Assistant Professor from Department of Biology. So you all know in our living cell have different kinds of cell organelles. Each cell organelle have its own specific functions. Cell organelles such as lysosomes, endosomes and microbodies have its own functions, its own structure. So let us discuss about first of all lysosomes. Lysosomes are vesicular, membrane bound and enzyme filled cell organelle concerned with the intracellular or extracellular digestion. They were isolated and named lysosome by Christian D. Dubé in 1955. They formed from Golgi bodies. It's present in most eukaryotic cells, but in prokaryotes, it's surrounded by extracytoplasmic lysosome. It's absent in erythrocytes. So next, we have to discuss about lysosome structure and chemistry. In this picture, we have labeled that about it's have a membrane, lipid bilayer and glycosylated membrane transport proteins and also hydrolytic enzyme mixture. So let us discuss about its structure in detail. Lysosomes are rounded or globular sacs filled with hydrolytic enzymes. They have capability to break down many types of biomolecules and have a pH 5. They enveloped by single membrane. It's mostly micellar with lipoprotein composition. The membrane protein are glycosylated to protect themselves from degradation by lysosomal proteases. Lysosome suicide bags. The membrane contains substance called stabilizer, which make it stable and impermeable to lysosomal enzyme. They include cholesterol, cortisol, heparin, etc. Certain other substances can destabilize the membrane and make it permeable to lysosomal enzymes. They are called labilizers. They include ubiquinol, estradiol, vitamins A and K. When the membrane gets destabilized or injured, the enzyme escape to cytoplasm. Normally, they may not be harmful to the cell since they may become inactive at the pH of cytosol, that is about 7 to 7.2. But sometimes they may digest the whole cell by virtue of this property called suicide bag. So next we have to discuss about lysosome polymorphism property. Lysosome exist in different forms at different stages of their life cycle because of their fusion with other structures for digestive purpose. First one, primary lysosome. These are the nascent lysosome. Second one, secondary lysosome. It's two type, heterolysosome and autolysosome. Heterolysosome, fusion of primary lysosome with endosome known as heterolysosome, and the fusion of primary lysosome with the cytosagrisome. It's known as autolysosome. And the third type is telolysosome, and also postlysosome is also present. The latest stage of lysosome is the telolysosome, and the remain inside cell has a degranulated bodies. Next, we have to discuss about its function. It's a intracellular digestion. It's involved in intracellular digestion and dissolution of cells, extracellular materials and larval tissues. Help in fertilization, autolysis in cartilage and bone tissues. Next, we have to discuss about The second cell organelle, endosome. Endosome or receptosome are membrane-bound cell organelle found within the cell. It's also called vesicle. It's formed from endocytosis. They are essential for the control of substance in and out of the cell. It helps in intracellular traffic and the sorting of ligand and receptors. It's a temporary vesicle for transportation. Endosomes are vesicles that pitch off the plasma membrane. Therefore, the lipid bilayer of endosomes contain plasma membrane proteins and the lumen contains extracellular components. Endosomes types. There are three different types of endosomes: early endosomes, late endosomes, recycling endosomes. They are distinguished by the time it takes for endocytosed material to reach them and by markers such as RABs. Endosome function. Early endosome is closed to plasma membrane and receives endocytosis vesicle. Late endosome is near to nucleus containing acid hydrolysis from Golgi body and send a vesicle for recycling to Golgi body. Recycling endosome is deeper in the cell and deliver vesicle to plasma membrane. So next we have to discuss about the third kind of cell organelle, microbodies. Microbodies are small membrane enzyme filled lysosome like cell organelle found in close association with the endoplasmic reticulum. They were discovered by Rodin in 1954. It's a round or elongated structure delimited by a single membrane and filled with a densely granular and enzyme-rich matrix. They are oxidative organelles actively involved in the breakdown of hydrogen peroxide and fats, oxidation of amino acids, fatty acid, and uric acid. Microbodies type. Based on the morphological features enzymatic composition, they were divided into three: peroxisome, microperoxisome, glyoxisome. Microbodies, peroxisome. Peroxisome, they were discovered by Dewey 1965. Or peroxisome are self-duplicating spherical or a dumbbell-shaped membrane-bound bodies. They have crystalline core that is contain oxidase and catalysis delimited by a single unit membrane. Animal peroxism is concerned with the oxidation of amino acids, uric acid, and generation and breakdown of hydrogen peroxide. Hydrogen peroxide is a strong oxidizing agent, potentially harmful to cell. The enzyme catalyzes the breakdowns. Catalyzer breaks it down to water and oxygen within peroxisome itself, and also beta oxidation of fatty acids. In plant peroxisome, it's the most found microbodies in the photosynthetic tissue of green leaves. They contain enzymes of glycolate. Microperoxisome. They were discovered by Novikov 1973. Found in mammalian tissue. It's a small sized crystalline core. It's a small sized one. Crystalline core are absent. Present oxidative and catalytic enzymes. They possess numerous slender connections with the smooth endoplasmic reticulum. Next, glyoxysomes. They were discovered by Breedenbench and Beavers in 1967. It's a highly specialized plant microbodies. They are concerned with the conversion of fatty acids to carbohydrates in glyoxylic acid cycle. They are roughly spherical organelles consisting of protein matrix and crystalline core delimited by a single unit membrane. Thank you.